Welcome back to the deep dive. Usually when we talk about human anatomy or biology, we think about it like building a house. You pour the foundation, you frame the walls, you put on the roof. It's a linear process.
Right, step by step.
But I've been reading through this stack of embryology texts you sent over, specifically looking at the urogenital system. And I have to say, the body does not build like an architect.
Oh.
How does it build?
It builds like a hoarder who is absolutely obsessed with recycling everything.
That is a very evocative way to put it. I like that.
I mean, seriously, I'm looking at the notes here, and it seems like we build things, use them for maybe a week, tear them down, turn the rubble into something else, or just leave the scaffolding up because, hey, we might need it later. It feels chaotic.
It does feel chaotic, but there is a profound logic to it. We call it the logic of transformation. And you're absolutely right to focus on that recycling aspect because the urogenital system is the best example of it in the entire human body.
Yeah. I mean, we are talking about two massive, completely distinct biological tasks here. Getting rid of liquid waste, the urinary system, and, well, ensuring the survival of the species, the reproductive system.
Two totally different jobs.
And the punchline of this entire deep dive is that nature uses the exact same box of spare parts to build both.
That's what we're digging into today, the paths of water and reproduction. And just to orient everyone, we are pulling from the heavy hitters. I've got Langman's Medical Embryology open here, which is just fantastic for those shifting timelines.
A classic.
I've also got I.B. Singh's Human Embryology, which I really like because he breaks down the structural sequences so clearly. It's very logical.
Very exam-oriented, which is helpful.
And then Moore and Prasad's The Developing Human, which is, and I know you love this one, it's so focused on the clinical reality, like what happens when this stuff goes wrong.
More is the so what book.
Yeah.
It always brings it back to the patient. But before we get lost in the weeds, I do want to reiterate the mission here. If you are a medical student listening to this or a clinician brushing up, or even just someone who's fascinated by how we get made, please don't treat this conversation as a replacement for the books.
Absolutely not. This is the ready reckoner. This is the synthesis. We want to give you the memory anchor so that when you go back and read Langman, the text actually makes sense. It clicks. But please go read the books.
Exactly. So where does this grand recycling project begin?
Well, looking at the map, and I'm trying to visualize the early embryo here, we are looking at the intermediate mesoderm. Can you place us in the body? Because I know we have the spine forming in the middle.
Right. So picture the embryo as a flat disc that is just folded into a kind of tube. Running down the back, you have the neural tube, that's the future spinal cord.
Okay.
Flanking that, you have the paraxial mesoderm, which breaks up into these blocks called somites. Those will become your vertebrae and muscles. Then if you move just a little bit further out to the side, between those somites and the lateral body wall, there is this longitudinal ridge of tissue. That's it. That is the intermediate mesoderm.
And this is the neighborhood where everything happens.
This is the common origin. Both the urinary system and the genital system arise from this specific strip of tissue. They are neighbors, they are roommates, and for a long time they are sharing the exact same plumbing.
Which leads to the first thing that really threw me for a loop in the reading. I.B. Singh talks about this concept that ontogeny repeats phylogeny.
Ah, yes.
Which is a bit of a tongue twister, but it basically means our own individual development replays our species' evolution.
A very simplified version of it, yes.
Yeah.
The embryo goes through stages that resemble the adult forms of its evolutionary ancestors.
And because of that, we don't just grow a kidney. We grow, is it really 3 of them?
We do. We cycle through 3 different successive kidney systems. It's like the body is upgrading its operating system in real time.
Okay, so let's walk through these because this is where the recycling theme really kicks in. The first one is the pronephros. And I'm looking at the diagram in Langman, and this thing it's in the neck.
It is, the cervical region.
Why on earth do we have a kidney in our neck?
Well, we don't, really. It appears right at the beginning of the 4th week. It consists of these 7 to 10 solid clusters of cells called nephratomes. In some primitive fish, this region actually functions as a kidney. But in humans, it's entirely vestigial. It is non-functional.
So it's a dummy run, a practice kidney.
You can think of it that way. By the end of the 4th week, it's mostly gone. But, and here's the first rule of embryological recycling, you can't just dismiss it. It has a crucial job.
Which is?
Even though the pronephros itself disappears, it lays down the track. It initiates the formation of a duct that runs from the neck down the body towards the cloaca, the common sewer.
Ah, so if the pronephros didn't start that duct,
the next kidneys couldn't develop. They wouldn't have a pipe to connect to. So the pronephros builds the highway and then immediately retires.
Okay, so then we get the second kidney, the mesonephros. Now this one looks a bit more serious.
It is. This appears late in the 4th week, moving down into the thorax and lumbar regions. This is the interim kidney. It's a large, elongated organ. It has glomeruli, it has tubules, it actually filters blood.
It makes urine.
It does. It produces a little bit of urine during the first trimester.
And for a few weeks, this is actually keeping us alive.
Well, the placenta is doing all the heavy lifting for cleaning the blood, but the mesonephros is definitely functioning. But its real value, its lasting legacy, isn't the urine it makes, it's the duct it uses.
The mesonephric duct.
Also known as the Wolffian duct. This is a name you need to tattoo on your brain for this entire deep dive.
Okay, Wolffian duct. Got it.
Because eventually, the mesonephric kidney, the actual filtration unit, degenerates. It dies off. But the duct, that tube remains.
And this is the part that blows my mind. In males, that drainage pipe for the temporary kidney, that becomes the vas deferens, the tube that carries sperm.
It does. It also forms the epididymis and the ejaculatory duct. Essentially, the core plumbing for the male reproductive tract is the repurposed sewer pipe of a kidney we discarded in the 2nd month of life.
That is just wild. Talk about efficiency.
It's opportunistic.
Yeah.
Nature hates to build something from scratch if there's a part lying around it can use, but we aren't there yet. We still need a permanent kidney because the mesonephros is going to fade away.
Which brings us to number 3, the metanephros.
The permanent kidney.
And this starts in the 5th week. Now, the way Langman describes this, it's not like the other two. It doesn't just appear. It seems like a collision or a merger between two different things.
It is, and this is a classic exam topic. We call this the dual origin of the kidney. The kidney isn't one continuous tissue that grows. It's built from 2 separate sources that have to find each other and shake hands.
The perfect handshake. I like that. So let's break down the 2 players. First up, we have the ureteric bud.
Right, so imagine you are standing inside that mesonephric duct, the Wolffian duct we just talked about, down near where it drains into the cloaca. Suddenly, a little spout, a little bud, grows out of the side of the wall. That is the ureteric bud.
And because it's a tube growing out of a tube,
it is destined to become the collecting system.
So this is the plumbing, the ureters that drain the kidney, the renal pelvis, the major and minor calyces, all the stuff that moves fluid.
Exactly. Think of it as the branching pipes of an irrigation system. But pipes are totally useless without a factory to fill them. That factory comes from the second source, the metanephric blastema.
Blastema always sounds like some kind of sci-fi weapon to me.
It just means a mass of undifferentiated cells, a metanephric mesoderm. This tissue comes from the intermediate mesoderm in the sacral region, so way down in the pelvis. It just sits there waiting.
Waiting for the ureteric bud to arrive.
Precisely. When the bud grows towards and penetrates this massive tissue, it triggers the blastema to form the excretory system.
The factory workers.
The nephrons. Bowman's capsule, the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule. These are the microscopic units that actually filter the blood and make the urine.
Okay, so let me see if I have this straight. The ureteric bud becomes the drainage pipes, and the metanephric blastema becomes the filtration factories.
You got it.
But the books, especially Moore and Prasad, they talk about this reciprocal induction. It sounds like they are shouting instructions at each other across a construction site.
It's a molecular conversation, and it's actually quite beautiful. It's a classic example of tissues needing each other to develop. The metanephric mesenchyme, the blastema, it releases a chemical signal called GDNF, glial-derived neurotrophic factor.
GDNF.
It's essentially screaming, come over here, I'm here, branch out.
And the bud hears this.
The ureteric bud has a specific receptor on its surface for GDNF called RETT. When GDNF hits that RE key receptor, it's like a key in a lock. It tells the bud to grow and branch. We call this branching morphogenesis.
So it's not just one pipe, it becomes a tree.
A tree with millions of branches. And as it branches, the bud sends signals back to the blastema saying, okay, you've called me over, now it's your turn. Stop being a loose pile of cells. Differentiate, turn into epithelium, make tubes, become nephrons.
So if they don't talk to each other, no kidney.
Precisely. If that handshake misses, if there's a mutation in the GDNF gene or the RET receptor is faulty, you get renal agenesis. The kidney just never forms.
And this leads directly to a clinical scenario that I found really disturbing in Moore and Prasad, the Potter sequence.
It is disturbing, but it's a powerful lesson in how interconnected every part of development is. It shows you that a kidney problem isn't just a kidney problem.
So paint the picture for us. What does a baby with Potter sequence look like at birth?
The classic Potter sequence presents with a flattened nose, a recessed chin, prominent folds under the eyes, low-set ears. It's often described as Potter facies or flattened facies.
Okay.
They might also have limb deformities, like clubbed feet. And most critically, they're born in severe respiratory distress. They can't breathe.
Okay, so help me connect the dots here. We just established this is caused by a missing kidney. How does no kidney lead to a flat face and bad lungs? It seems completely unrelated.
It's a hydraulics problem. Think about the baby in the womb. They're floating in a bag of water, the amniotic fluid. Where does that fluid actually come from?
I always just assumed it was mostly water from the mom's circulation or something.
In the very beginning, yes. But by the second half of pregnancy, the vast majority of amniotic fluid is fetal urine.
The baby is floating in its own pee.
It is. The baby swallows the fluid, it gets absorbed, processed through the kidneys, and then they pee it out again. It's a constant cycle that maintains the volume of fluid in the amniotic sac.
Okay, so if you have renal agenesis, no kidneys,
Right.
you have no urine.
Which means you have no amniotic fluid. The medical term for this is oligohydramnios. Oligo for few, hydramnios for the fluid.
Right.
Now imagine the uterus without that water cushion. The muscular walls of the uterus start to contract and shrink down, pressing directly onto the fetus.
Oh, I see. It's like being vacuum packed.
Exactly. The uterus physically compresses the fetus. That's what flattens the face and twists the feet into clubfoot position. But the lung issue is even more critical.
Why the lungs?
To develop properly, fetal lungs need to breathe that amniotic fluid in and out. The fluid pressure inside the developing airways is what expands them and allows the cells to proliferate and mature.
It stretches them out.
It does. Without that fluid, and with the chest being physically crushed by the uterine wall, the lungs remain tiny and underdeveloped. They're hypoplastic.
So the baby dies not from kidney failure, but from lung failure.
Correct. The cause of death in Potter's sequence is pulmonary hypoplasia. It's a powerful lesson. The kidney isn't just a waste organ in utero. It's a structural engineer that maintains the living space for the fetus, which is essential for other organs, like the lungs, to grow.
That is heavy, but a really clear connection. So let's assume the handshake works. We have kidneys, but they start in the pelvis, right? The sacral region.
Yep, down in the basement.
But my kidneys are up under my ribs, so they have to move. This is the ascent of the kidneys.
Between the 6th and 9th weeks, they migrate superiorly. Well, migrate is a bit of a misnomer.
Oh.
It's more that the caudal end of the body, the butt and legs, grows away from them so rapidly, and the overall curvature of the fetus straightens out.
Yeah.
So, relatively speaking, the kidneys end up much higher in the lumbar region, around T12 to L3.
And as they move up, they have to change their power supply.
The blood supply, exactly. This is like climbing a ladder. When the kidney is in the pelvis, it grabs a branch from the common iliac arteries. As it moves up, it lets go and grabs a new, higher branch directly from the aorta. Then it moves higher and grabs another one.
Does it let go of the old ones cleanly?
It's supposed to. As it establishes a new, higher connection, the lower vessel should degenerate and disappear. But, going back to our theme, the body is a hoarder. It often forgets to delete the old files.
So you can have extra arteries.
Very common. About 25%, maybe even 30% of the population has accessory renal arteries. These are just the old lower vessels that never went away.
Is that a problem for the person?
Usually not. The kidney gets plenty of blood. But it's a huge deal for a surgeon.
Yeah.
If you go in to do a nephrectomy or a transplant and you clamp what you think is the renal artery, but you miss an accessory one hiding somewhere, you're going to have a lot of bleeding.
I can imagine. What about when the ascent itself gets physically blocked? I saw a diagram in Langman of a horseshoe kidney.
Yes. A classic anomaly. Sometimes, when the two kidneys are still down in the pelvis, their lower poles are so close that they touch and fuse together. So instead of two separate beams, you have a U-shape, a horseshoe.
And that U-shape causes a problem when they try to go up.
Right. They start their ascent, but then thunk. They hit a ceiling.
What's the ceiling?
They get hooked around the inferior mesenteric artery. That artery comes off the front of the aorta to feed the large intestine, and it forms a physical barrier. The fused kidney can't pass it. So people with a horseshoe kidney have it located much lower in the abdomen than normal, draped over the aorta.
Fascinating. So if you see a kidney in the wrong place on a scan, an ectopic kidney, you have to think about the arteries.
Always. Anatomy is just developmental history.
Let's follow the plumbing down. We need to talk about the final drain, the cloaca.
The cloaca is the Latin word for sewer. In the early embryo, just like in birds or reptiles, we have one common chamber for everything. Urine from the urogenital system enters it, and feces from the hindgut enters it.
Not a great design for a civilized mammal. We like to keep those separate.
So we build a wall. A septum of tissue called the urorectal septum grows down like a curtain and splits the room in two. The front room becomes the urogenital sinus, which will form the bladder and urethra. The back room becomes the anorectal canal.
There is one detail about the bladder that I found really confusing in Langman. The trigony. It says the bladder is endodermal, which makes sense. It comes from the gut tube, basically. But the trigony is mesodermal. How does that work?
This is one of those beautiful aha moments for understanding adult anatomy. The trigony is that smooth, triangular patch at the base of the bladder, where the two ureters enter from the top corners and the urethra exits from the bottom.
Right. I remember that from anatomy lab. It looks different from the rest of the bladder wall.
And it is different because of its origin. Embryologically, that triangle is actually formed by the absorption of the lower ends of the mesonephric ducts.
The Wolffian ducts again?
The Wolffian ducts again. As the bladder grows and expands, it incorporates the ends of those ducts into its back wall. The ureters, which originally butted off the Wolffian ducts, get their own separate entrances.
So the trigone is essentially a patch of the old kidney duct that got plastered onto the bladder wall.
Exactly. It's a patch of mesoderm that gets incorporated into an endoderm bag. Eventually, the endodermal lining grows over it, but the underlying structure and its nerve supply is different because of that origin. It's a ghost of the mesonephros.
Okay, let's shift gears. This is a huge transition. We've built the plumbing for water, we have functioning kidneys, ureters, a bladder, but now we hit the part of the outline that I think is the most surprising, the indifferent stage of the genital system.
This is weeks 1 through 6, and really up to week 7.
If I'm looking at an embryo at week 5, can I tell if it's a boy or a girl?
Morphologically, absolutely not. You cannot.
That's wild. We are physically identical at that point.
We have the exact same starting ingredients. We have these thickenings of tissue called the gonadal ridges, which will become either testes or ovaries.
And the actual sex cells.
Here's a fun fact about the ingredients. The primordial germ cells. The cells that will eventually become sperm or eggs, they aren't from around here.
What do you mean?
They don't originate in the embryo's body proper. They originate in the wall of the yolk sac, outside the embryo.
They're immigrants.
They are. In the 4th week, they begin this incredible migration from the yolk sac, crawling like amoebas along the dorsal mesentery of the gut, and they invade the genital ridges. If they don't make that journey, the gonads never develop properly.
Wow. So we have the gonads waiting for the seeds to arrive, and then we have the ducts. And this is the definition of hedging your bets.
It is. At this stage, every single embryo, XX or XY, has both sets of pipes. We have the mesonephric ducts, the Wolffian ducts, which are the potential male system.
The ones we just talked about.
And running right next to them, we have a second pair of ducts, the paramesonephric or Mullerian ducts. These are the potential female system.
We are double plumbed. Everyone is double plumbed.
Everyone starts with the capacity to build either system. We are prepared for either outcome.
So what flips the switch? What's the deciding factor?
It all comes down to a single gene on the Y chromosome, a gene called SRY,
SRY.
sex-determining region on Y. This gene produces a protein called TDF, testis determining factor.
It sounds like a boss. I am the test determining factor.
It absolutely acts like one. If TDF is present, it commands the primitive sex cords in the gonad to penetrate deep into the medulla of the ridge and organize into a testis.
And if it's not there?
If SRY is absent, if you are XX, that command never comes. The cords remain in the cortex, the outer layer of the gonad, and they eventually differentiate into an ovary.
Langman makes a point here that I want to clarify. It says female development is the default, which can sound a bit passive.
It's a complicated concept. The development of the ducts is passive in females, which we'll get to. But forming a fully functional ovary isn't just not forming a testis. You need 2 X chromosomes.
Yeah?
We know this from Turner syndrome, where you have a 45XO arrangement. Without that second X, you don't get functional ovaries, you get non-functional streak gonads. So you need the double X to maintain the ovary. But for that initial switch, yes, SRY is the master key that turns development down the male path.
Okay, so let's say the key is turned. SRY is present. A testis forms. Now the battle of the ducts begins.
It's a war on 2 fronts. The newly formed testis has to do 2 things simultaneously: build the male house and tear down the female house.
So who does the building?
The Leydig cells in the testes. They start pumping out testosterone. Testosterone finds those mesonephric or Wolffian ducts, remember the old kidney drains, and it stimulates them. It essentially tells them, don't die, I need you. turn into the epididymis, turn into the vas deferens, turn into the ejaculatory duct.
So testosterone saves the male pipes from degenerating. Who destroys the female pipes?
The Sertoli cells in the testes. They produce a completely different hormone called AMH, anti-Mullerian hormone.
Does exactly what it says on the tip.
Exactly. AMH, which is also called Mullerian inhibiting substance, or MIS, attacks the paramesonephric or Mullerian ducts. It causes them to rapidly regress and disappear. So the male embryo actively destroys the potential to form a uterus and fallopian tubes.
That's fascinating. It's an active demolition project.
Very active.
Now let's flip it. What happens in the female embryo? No Y chromosome, no SRY, no testis.
Right. So no Leydig cells, which means no testosterone.
And the use it or lose it principle applies here.
Big time. Without testosterone stimulation, the Wolffian ducts, those male pipes, just wither away. They have no support, so they're gone.
And because there are no Sertoli cells?
There is no AMH. So the paramesonephric or Mullerian ducts are safe. In fact, in the presence of estrogen from the mother and the placenta, they survive and thrive.
And they do something really cool. They fuse.
Yes. This is a key difference from the male side. The two Mullerian ducts run down the sides of the body. At their cranial or upper ends, they stay separate. These become the fallopian tubes or uterine tubes.
Okay.
But as they move down into the pelvis, they swing toward the center and they zip together in the midline.
Like zipping up a jacket.
That's a perfect analogy.
Huh.
They fuse to form a single tube called the uterovaginal canal. This structure becomes the uterus and the upper part of the vagina.
This visual of zipping up really helps explain the anomalies that Moore talks about, because if the zipper gets stuck,
You get fusion defects. If they don't fuse at all, if the zipper never starts, you get uterus didelphys, a complete double uterus, two separate uterine horns, two cervices, and sometimes even a divided vagina.
and if they zip up only part of the way.
You might get a bicornuate uterus. It looks heart-shaped. The bottom part is fused, but the top splits into two distinct horns.
Does that matter for the patient later in life?
If it certainly can.
Yeah.
These uterine anomalies are often associated with fertility issues, recurrent miscarriages, or problems during pregnancy because the shape of the uterine cavity is abnormal. It's a direct anatomical echo of that embryological moment where the two tubes met in the middle.
Okay, so we've sorted the internal plumbing, we've got a uterus, or we've got a vas deferens, but now we have to decorate the exterior. And honestly, this is the part of the chapter where I had to stare at the pictures for a long time, because the transformation of the external genitalia it's like origami.
It is. And once again, we start within a different stage. From week 3 to week 6, it all looks the same. You have a bump at the top called the genital tubercle. You have two folds of tissue below it called the urethral folds, and two larger swellings on the outside called the labioscrotal or genital swellings.
And everyone has this exact same setup.
Everyone, boy, girl, it looks identical.
So what changes it? What's the signal?
Hormones again. Specifically, a very potent version of testosterone called dihydrotestosterone, or DHT. An enzyme converts testosterone to DHT in these tissues.
So in the male embryo, DHT hits these structures. What happens?
It causes two things: growth and fusion. The genital tubercle undergoes rapid elongation to become the glans penis. The urethral folds, which start out as the open lips of a groove, start to zip up from the back to the front. They fuse together to enclose the urethra inside the shaft of the penis.
So the open trench becomes a closed tube.
Correct. And the genital swellings, those outer bumps, they migrate towards the middle and fuse to form the scrotum, creating the bag that the testes will later descend into.
Okay, now let's look at the female. No testes, so no DHT.
In the absence of DHT, there is minimal growth and no fusion. The genital tubercle becomes the clitoris. The urethral folds remain open, they become the labia minora, and the genital swellings remain separate. They become the labia majora.
This is why we say they are homologous structures. They come from the exact same starting material.
Yes, the scrotum is the labia majora, just fused. The skin on the underside of the penis, the ventral aspect, is the labia minora, just zipped up. The penis is the clitoris, just enlarged.
That explains hypospadias then. I've heard of this. It's pretty common, right?
Very common, about 1 in 300 male births.
This is where the opening of the urethra is in the wrong place.
Right. Instead of opening at the tip of the glans penis, the urethra opens somewhere on the underside, the ventral surface of the penis. It can be near the tip, along the shaft, or even down by the scrotum.
And the cause is embryological.
It's a direct failure of that zipping up process. The urethral folds failed to fuse completely. If the zipper stops halfway, you're left with a hole there. That's the hypospadias.
Wow, okay. We can't talk about the male system without talking about the big commute. The testes don't stay up in the abdomen where they form.
The descent of the testes. This is a long and perilous journey. The testes develop way up by the kidneys in the lumbar region. They have to get all the way down to the scrotum.
How do they know where to go? Is there a map?
They are towed. There is a fibrous cord, a ligament called the gubernaculum.
Gubernaculum. Best word in embryology, hands down.
It sounds like gubernatorial, like a governor. It actually comes from the Latin word for helm or rudder.
Yeah.
It steers them. The gubernaculum is attached to the bottom of the testes and to the floor of what will become the scrotum.
And it pulls it down.
As the fetus grows, this ligament relatively shortens, and it guides the testes through the abdominal wall, specifically through the inguinal canal and into the scrotum. This usually happens in the last trimester.
And it drags some layers of the abdominal wall with it.
It does. It drags a finger-like projection of the peritoneum, the lining of the abdominal cavity, called the processus vaginalis.
And that tunnel that it creates is supposed to close up, right?
It absolutely must close. If it stays open, you have a direct highway from the abdomen to the scrotum. That's a congenital inguinal hernia. Loops of intestine can fall into the scrotum.
Oh.
Or if the hole is just tiny, abdominal fluid can trickle in, and you get a fluid collection around the testes called a hydrocele.
Now, wait a second. Females have a gubernaculum too, don't they?
We do, but the ovaries don't need to go all the way to the labia. They descend out of the abdomen and into the pelvis, and then they stop.
Yeah.
They get hung up on the uterus, which is formed in the middle of their path.
This is what happens to the toe rope, the gubernaculum.
It gets attached to the uterus and effectively gets split in two. The upper part becomes the ovarian ligament, holding the ovary to the uterus. The lower part continues on through the inguinal canal and becomes the round ligament of the uterus, which attaches to the labia majora.
So when you're in surgery and you dissect the round ligament, you are holding the female equivalent of the testis's guide wire.
You are holding the ghost of the gubernaculum.
I love those connections. Now, I want to spend the last chunk of our time on something really important and something that can be confusing. The disorders of sex development, or DSDs. This used to be called intersex, right?
Yes. The terminology has shifted to DSD to be more clinically precise and less stigmatizing. And Moore and Prasad really emphasize that this is a spectrum. It happens when the chromosomal sex, the gonadal sex, and the external phenotypic appearance don't align in the typical way.
And this is the part of the reading that really challenged my simple understanding of how sex works. I want to walk through two specific scenarios because they explain the underlying mechanisms so well. Let's start with androgen insensitivity syndrome, or AIS.
Formerly called testicular feminization. And this is the classic example of a 46XY DSD.
Okay, so let's set the scene. Imagine a teenage girl. She looks female, she was raised female, she identifies as female. She goes to the doctor because she's 16 and still hasn't started her period. She has primary amenorrhea.
A common clinical presentation.
The doctor does an ultrasound and is surprised to find there's no uterus.
And then they do a karyotype, a genetic test, and find that her chromosomes are 46XY.
How is that possible? She is genetically male but looks completely female.
Let's go back to our developmental pathway.
Mm-hmm.
This individual has testes. They formed correctly because she has the SRY gene. These tests are usually undescended, hidden inside the abdomen.
And they're making hormones.
Yes, the Leydig cells are working overtime, pumping out massive amounts of testosterone. So she has the signal, but she lacks the receiver. There is a mutation in the androgen receptor gene. The cells of her body, the genital tubercle, the urethral folds, the labioscrotal swellings, they cannot hear the testosterone. It's like shouting at someone who's wearing noise-canceling headphones.
So the body thinks no testosterone signal, must be female.
Exactly. The external genitalia develop along the default female path. You get a clitoris, labia, and a short blind-ending vaginal pouch.
But why no uterus? This is the key part.
Remember the other hormone the testes make. The Sertoli cells are functioning just fine. They produce anti-Mullerian hormone, AMH.
And the body can hear AMH.
Yes, the receptors for AMH are totally fine. So the AMH does its job and destroys the Mullerian ducts. No uterus, no fallopian tubes. So you end up with a person who looks female externally, but has no internal female organs and has hidden tests.
That is a perfect, if mind-bending example of how genetics isn't destiny hormones, and crucially, their receptors are.
It is.
Now, flip the script. 46XX-DSD. The most common cause is congenital adrenal hyperplasia, or CAH.
This is the most common cause of ambiguous genitalia in newborns.
So, scene, a baby is born. The delivery team looks at the baby and says, it's a boy, I think. The clitoris is very large, looking like a small penis. The labia are partially fused, looking like a scrotum. but a chromosome test shows this is a 46XX female.
Right. So what happened here? The baby has ovaries. They formed correctly.
Mm.
The baby has a uterus. That also formed correctly. But the baby also has adrenal glands, which sit on top of the kidneys.
And hyperplasia means they're overgrown.
Yes. Due to a common genetic defect, a missing enzyme, usually 21-hydroxylase, the adrenal glands can't make their normal hormones, like cortisol. So the brain sends a strong signal to the adrenals, work harder. In trying to work harder, they get huge, and they dump out tons of precursor molecules. Specifically, androgens, male hormones.
So this female fetus is bathing in a sea of testosterone-like hormones from her own adrenal glands.
Exactly. And unlike the previous case of AI, her androgen receptors work perfectly fine. So this flood of adrenal androgens hits the external genitalia and masculinizes them. It enlarges the clitoris, it fuses the labia.
But internally...
Internally, she is female, she has no testes, so no AMH was ever produced. Therefore, her Müllerian ducts were never destroyed. She has a completely normal uterus, fallopian tubes, and ovaries. It's just the exterior that has been modified by this abnormal source of hormones.
It really drives home how distinct the development of the internal ducts and the external genitalia are. They're two separate stories.
Two separate stories, run by two different hormones from the testes. Testosterone for the Wolffian ducts and AMH for the Müllerian ducts. It's a beautiful, logical system.
So we've gone from 3 kidneys in the neck to a single permanent kidney in the abdomen. We've gone from an indifferent unisex starter pack to fully differentiated male or female anatomy. What is the big overarching takeaway for you here?
For me, it's the fragility versus robustness paradox. We've talked about so many things that can go wrong. Potter sequence, hypospadias, uterine anomalies, DSDs. The whole process relies on this precise, timed cascade of genes, WT1, SRY, GDNF, and this perfect hormonal symphony.
Right.
One receptor fails, one fold doesn't zip, one artery doesn't disappear, and a whole plan shifts. It feels like walking a tightrope over a canyon.
It does. It feels incredibly fragile.
It is, and yet the vast majority of the time, it works flawlessly. We come out with 2 functioning kidneys, a working bladder, and distinct functional reproductive tracts. There's a profound robustness built into this fragile system.
And for the learner listening to this, what's the one thing they should take with them?
I'd say, look for the ghosts. When you study adult anatomy, don't just memorize the structures as they are now. See their history. When you see the trigony of the bladder, tell yourself, that's the ghost of the mesonephric duct. When you dissect the round ligament of the uterus, say, that's the ghost of the gubernaculum.
It makes the anatomy 4D, adding the dimension of time.
Exactly. Every structure tells a story of its own development.
I love that. Well, that wraps up our deep dive into the paths of water and reproduction. My advice for you listening, open Langman or more right now. Find the color-coded diagrams of the urogenital sinus and external genitalia differentiation. Visualize that purple merging with that yellow, that fold zipping up. That's how you lock this in for good.
Don't just read it. See it happening in your mind's eye.
Thanks for listening. We'll catch you on the next deep dive.